Hey guys, this is Mr. Mahmood, and you're watching Division Lecture 3. This is the final lecture of the division unit. The first couple of lectures have introduced you to the structure of DNA and the importance of the cell cycle in terms of what we consider interphase, which is the majority of the cycle, and the DNA replication that happens there. And it all leads up to this little tiny time interval within the cell cycle where a whole lot happens. And we call that final little cycle the M phase, or mitosis. And within mitosis, a lot of different things are going to happen in order to allow the cell to divide. And it divides through a process that we consider asexual reproduction. So before we start talking about mitosis in detail, we'll talk about what asexual reproduction is, what some major advantages of asexual reproduction could be, and some specific examples of types of asexual reproduction that organisms go through throughout all 6 kingdoms. Then we'll really get into the detail of how mitosis happens, all the specific intervals and steps that happen in between that you need to make sure you know well. Talk about the final step of splitting that cell after we've gone through mitosis, which is called cytokinesis. And then we're going to talk about a very serious set of disorders that all derive from problems with the cell cycle, and these are what we call types of cancer. So I'm sure you've heard the term cancer before, but you didn't really understand what it means. We're going to really understand exactly how it relates to this exact process of the cell cycle. Alright, so before we jump into any specifics about what mitosis actually is, we first need to talk about the type of reproduction that's going on here. You guys remember one of the 8 characteristics of life is that all living things reproduce. Now, there are 2 basic categories of reproduction. I think we've introduced this before. Asexual reproduction compared to sexual reproduction. To define asexual reproduction, it's usually 2 specific characteristics that we talk about. One is the number of parents. And in asexual reproduction, we always begin with 1 parent. So asexual reproduction always starts with 1 parent. And in terms of the offspring that are produced, the offspring that are made are usually identical to each other and identical to the original. So if we're talking about 1 parent producing identical offspring, that's usually considered a type of asexual reproduction. But what are some advantages to asexual reproduction over sexual reproduction? Besides not having to pay for dinner and a movie, right? Basically, asexual reproduction is all about making exact copies. So if we're talking about wanting to make sure one generation after the next is always working the exact same way, this process of asexual reproduction makes sure that it happens. And we've talked about how important DNA replication is in allowing this asexual process to occur. If I want 2 cells to be produced that are identical to each other, identical to the original, I have to make sure that the DNA that goes into each one of those 2 new cells is also completely identical. So one huge advantage for asexual reproduction is to continue making exact copies, to make identical replicas of the original parent over and over again in order to maintain that homeostasis that needs to happen within every generation within any given tissue. It's very important that the same cells are being produced over and over again with almost no change whatsoever. That's very important for the functioning and the success of a cell in its homeostasis. Another big advantage to the cell division process that we've talked about before is due to size. Because of the cell cycle, gradually the cell getting bigger and bigger, all cells grow and develop, it gets to the point where it's so large that it's no longer efficient. It's taking too long to get where it needs to go, like driving through Texas, right? So it's very important, if we want a cell to remain efficient, it needs to split and divide back into 2 small pieces, which is another major advantage of asexual reproduction. It takes that big piece and splits it into 2 small pieces. And probably the most important advantage of asexual reproduction as compared to sexual, which we'll talk about in the next unit, is speed. Asexual reproduction can happen relatively quickly because there's no need for a process of 2 parents finding each other and going through the fertilization process or anything like that. It can divide itself as long as everything's ready for it to occur. Alright, so we know why cells divide asexually. Now let's talk about some of the basic categories of asexual reproduction. If we're talking about a eukaryotic cell, like animal cells and plant cells, protists and fungi, all the cells that have a nucleus, the process of division is going to involve mitosis, which by definition is nuclear cell division. So when we talk about mitosis, we're talking about dividing a cell that has a nucleus. So this is how eukaryotic cells divide. For other types of organisms, specifically prokaryotic, like bacteria, they go through a process called binary fission. Now, in diagram, it's very, very similar to mitosis. It's genetic information replicating itself first, so the DNA line goes through replication, doubles its number, then it basically separates itself out evenly, so you have equal amounts and equal information on either side of the cell, and then it just kind of splits in the middle and splits into two. It's called binary fission for a reason. Binary means two, fission means to separate. So binary fission by definition is one cell splitting into two, which is again pretty much what's going to be happening in mitosis, except in mitosis there are a lot of intermediate steps that go into play because it's a eukaryotic cell, so it's a lot more advanced. But bacteria go through this binary fission process. One of the sources of evidence behind endosymbiotic theory is the fact that both mitochondria and chloroplasts divide this way. They go through their own binary fission process within the cell in that G2 phase when they're getting prepared for the real cell division process that's going to happen in mitosis. Another way to think about this, there are basically two categories of students, right? Let's say we have the first category of student, which is highly organized, very responsible, and plans ahead. They know they have, say, a major project that's going to be due in a couple of weeks. So they decide they're going to look at their schedule, they're going to plan out their schedule so that every single night they take care of a little part of the project. So by the end of it, they can just accumulate it and put it all together and not have to really stress over it the night before. Right? That's much like binary fission, a whole lot like mitosis. There's a whole lot of work that's getting put in early and consistently to get the cell ready. And then once it's cell ready, the splitting process is pretty straightforward. Now let's think about the type of student that the rest of you fall into, which would be the ones that don't really plan ahead very well. They know about the project coming up just as well as the first group, but instead of trying to plan ahead and work out times every single night to get certain things done, they decide to push it off until the latest time possible, and then they freak out about it because it's due the next day or it's due very soon after. These guys are a whole lot like this next type of asexual reproduction. This process is what we call budding. This basically is a type of cell that rather than preparing ahead of time for division, it just starts a kind of a growth on the surface of the first cell. So you have a cell that's functioning properly, and all of a sudden a little growth will start to form off on the surface of one of these structures. And that growth starts to get gradually bigger and bigger and bigger. Think of it like a pimple that just never stops growing. And as it's growing, it's picking up all of the components that it needs and making more of it inside of itself, replicating its DNA as it goes, until it finally gets all the basic components it needs to be able to function on its own, and it just kind of pinches off and becomes its own cell. This process is called budding. This is a whole lot like that second group of student that doesn't really plan ahead very well. They just kind of do things as they go and gradually progress, not really worrying about planning and preparation. And then once they've gotten everything they need, they just kind of pinch off and become their own cell. This is a good example of something that yeast go through. Yeast is a type of fungus that divides through budding. Again, this is asexual because we're starting with one parent and producing an offspring that's eventually going to be identical to that original parent. But it's not preparing ahead of time like you see here in binary fission. So you have binary fission, prep first, then the split. You have budding, where it's just a growth and it kind of works as it goes, develops everything it needs as it's splitting until it breaks off. And then we get into some of the larger scale examples of asexual reproduction, where really we're talking about mitosis happening in all of these, because they're both examples of either animals or plants. This third example of asexual reproduction or division is something called regeneration. Now this is something that only certain species are capable of doing, and there are specific species of animals that can do this as well, including all categories of lizards. So if you were to break off a segment of this lizard that's not vital to their survival, so it doesn't have vital organs, like you can't chop a lizard down the middle of its torso and expect it to survive, but if you chop off its tail or it gets stuck or damage its tail and the tail breaks off, it can actually regenerate that tail. And you see that picture happening here. This type of lizard is actually starting to regrow a broken tail there at the end. This happens also with starfish and some other examples of animals where a part of the body that does not contain vital organs can actually regenerate itself. And this again is an example of asexual reproduction. It's just making identical copies of what's originally there, and you actually end up with something pretty similar to what you started with. Now I should warn you, we as humans do not have this trait. Don't go off to the kitchen and test it by chopping off your finger and hoping for the best. It won't grow back. Okay, we don't have this trait. This trait, for whatever reason, never became a necessity for our species, and we haven't developed this throughout us. But some organisms are able to go through this regeneration process that's also asexual. And then finally, plants go through a very unique process called vegetative propagation. Propagation just basically means making more or producing new types. In vegetative propagation, within one large plant, you can actually start producing newer, smaller plants off of that one large plant. And that newer, smaller plant will eventually just kind of fall off and continue to grow on its own. It's kind of like budding, but on a much, much larger scale. We're not just talking about one cell and something growing off of one cell. We're talking about a whole organism, and little tiny new organisms can develop on the surface of that other organism. And this happens kind of at the end of the stem of a plant, and because of some unique hormonal production that happens there. You see here, it represents the new development of multiple plants with their roots on the surface of another plant, and it'll actually fall off. Those roots, if they manage to get dug into the ground, can actually grow into an entirely new plant. So this again is called vegetative propagation. It's asexual reproduction because you're just starting with one parent and then you're producing an offspring from it. These offspring are identical to the original parent. All of these are examples of identical reproduction. So when we talk about mitosis, we're going to focus in on the same basic process, except here we're talking about eukaryotic cell division. And I keep stressing eukaryotic because the biggest problem here is you want to take DNA that you've replicated, so there's two identical copies, and you want to separate them to two equal sides of the cell so that it can split down the middle and become two new cells. But here's the basic riddle. How do you take a cell with a nucleus and get the DNA to separate evenly on both sides of the cell when the DNA never is allowed to leave a nucleus? Well, the answer is you can't. So what you have to end up doing is actually get rid of the nucleus through this process of mitosis, and then you have the genetic information that can now move where it needs to go. So it's very important that you recognize when we talk about mitosis, we're not really talking about cell division, we're talking about nuclear division. Basically the division of the information inside of that nucleus, which has been replicated after the S phase of mitosis. So we're now past all the parts of interphase. We're going to focus in now on that little tiny time frame right there at the end of the cycle that we consider the M phase or mitosis. So again, let's reverse a little bit, go back to the S phase. In DNA replication, remember we took the DNA line, the human DNA line, and we replicated it. We went through all the steps and talked about helicase and polymerase and all that stuff, right? So we've taken the whole DNA line and now made two identical copies of it. Now the reality is if I were to take the human DNA line, which is about 6 billion bases long, and then try to go through the division process with one strand starting at the top on the right and the other strand starting at the bottom on the left, 6 billion bases away, and then slowly working their way through, it would take an incredibly long period of time until the whole DNA line was finally replicated. So to become much more efficient, the cells have learned to take the human DNA line and break it into segments. And then each segment starts on one end and works their way through. This makes things a whole lot faster. The human DNA line is actually broken into 23 different segments when it goes through this replication process. And then each of these 23 different segments are then going to go through the replication process. So one side here, which represents one line of DNA, will go through the whole replication process and become two identical copies of the same DNA line. So what we have here are two identical copies of a certain segment of DNA held together in the middle by this protein structure. What you see here is called a chromosome. So the basic structure of a chromosome can be split into basic parts. Each side of a chromosome, remember, is one identical part of the DNA line. Each side of the chromosome is known as a chromatid, right? So you have two identical chromatids that are held together to make the chromosome. A lot of times, for whatever reason, scientists use the female representation when we're talking about relation between cells here. We're going to often refer to daughter cells as the offspring. And in this case, we're going to talk about the chromatids being known as sister chromatids because they're identical to each other, because of their relationship to each other. So a chromosome is actually made of two sister chromatids held together in the middle by this protein structure, this kind of like a big lasso holding it all together. That protein structure is known as a centromere or a kinetochore. If you hear either one of those, it basically refers to this protein that holds the two sister chromatids together in this puffy Cheetos shape. So this is the basic structure of a chromosome. It's actually two sister chromatids held together in the middle by the centromere. I know it looks like an X, but don't think of it like this, because in reality, it's two identical strands that are just held together in the middle here. So think of it more like two puffy Cheetos held together in the middle, because that's what it is. Now you remember, we've kept referring to human cells as having 46 chromosomes, right? Well, as you're going to learn later, it's because you have 23 chromosomes that came from your mother and 23 chromosomes that came from your father. So the combination of the two 23 chromosomes that you have from both mom and dad will give you your 46 chromosomes. So even though the human DNA line is 23 segments long, every single body cell that makes up who you are actually has two lines of DNA, one from mom, one from dad. So since each of them have 23 segments, that's where you get your 46 chromosomes. So they're chromosomes before they've ever really replicated. But once they replicate, now they have the structure that you're used to seeing. So this is after DNA replication. You're left with 46 of these chromosomes that each have two copies of the original information. So the ultimate goal of mitosis is going to be to make sure that each of those 46 chromosomes splits evenly so that one identical copy goes one way, the other identical copy goes the other way. And then when you make two new cells out of it, each new cell now has the exact same 46 segments that the other one has. So let's focus in now. Fast forward again and past G2, where these chromosomes are still in a nucleus, and they've now gone through all the replication process. G2 has gone through the remaining parts of preparation for cell division, which includes making more of the organelles, the mitochondria and the chloroplast going through their own binary fission. Building these little yellow things, you may remember talking about them at the end of the last video. These are called centrioles. They're going to play a critical role in the actual process of mitosis here. We're going to talk about centrioles quite a bit as we progress through each one of these phases. So we're now ready to end G2 and start the actual process of nuclear cell division, which we consider to be mitosis. So again, the biggest problem here is I want to get the DNA that's inside of that nucleus separated evenly to both sides of the cell so I can make two new cells, but the DNA won't leave the nucleus if the nucleus is still put together. So the very first step of mitosis, which we consider prophase, actually involves that nucleus breaking apart and dissolving. So this is the only time in a cell cycle where I can actually tell you a eukaryotic cell does not have a nucleus, and it's because you can't have a nucleus if you want to split everything up evenly. So this process of mitosis starts with prophase, where the nuclear envelope will begin to break apart. These chromosomes will really start to compact and condense, because it's going to be very important that they're really densely packed when the splitting process starts to happen. If they're really loose, then things wouldn't happen very consistently. So in preparation, all those chromosomes really coil up and condense and get very thick so that the separation can become a much smoother process. So this is what we consider prophase, the first phase of mitosis. There are four phases that we're going to go through. Those centrioles that we talked about have a very strong negative charge. So just like two magnets, if you have two magnets that are both the same charge and you have them close to each other, naturally they're going to start to separate away from each other. So in G2 they're made in the same place, so they start all in the same place, but once you let them kind of work naturally, they're naturally going to start to repel each other. So they start moving to opposite ends of the cell, and it's a circular cell, so that means they're pretty much just going to move to the opposite ends of the cell. Those opposite ends that they move to are what we're going to call the poles of the cell, kind of like the north and the south pole on Earth. It's just an even separation. So in prophase, nuclear envelope breaks apart, chromosomes condense and compact tightly, and the centrioles begin to move to the poles. Now when the centrioles move all the way out to the poles, remember they're very strongly negatively charged, so they're basically as far away from each other as they can be. And when the nuclear envelope finally begins to break apart, the DNA and all of the chromosomes that are inside of that nucleus are going to be able to move anywhere where they want to go. They're going to be able to actually freely move throughout the cell. But your DNA is also very strongly negatively charged. So it's a strong negative charge in your DNA as well. So you have two very strong negative charges on the two poles and another very strongly negative charged molecule that's trying to move anywhere within the cell. So the DNA and the chromosomes are going to try to move as far away from the poles as possible because they're repelled by the same charge. So if you're thinking about the Earth, if I want to get as far away from the North and South Pole as I possibly can, where would I go? Ecuador, right? I'd shoot right to the middle, right to the equator. And by doing that, I am equidistant from both poles. I'm as far away from the poles as I can be. That's exactly what's going to be happening to your chromosomes here in the next phase. So as the chromosomes are no longer constricted by a nucleus and the centrioles are off on either end of the cell on these poles, the chromosomes are naturally going to get as far away from the two poles as possible, which means they're going to start to line up right along the middle of the cell. And this phase is what we consider metaphase. The prefix meta literally means middle. So in metaphase, because the chromosomes are now free moving, the nucleus is gone, and the centrioles are so strongly negative on either side that the chromosomes get as far away from the sides as they can, they all start to line up right in the middle of the cell. So now we have all 46 of your chromosomes lined up one on top of the other in a straight line, basically, right along the middle of the cell. It's in this phase that also the centrioles will start releasing these little fibers called spindle fibers to attach to either side of the chromosome. Now, I want you to think of this sort of like fishing. Even if not all of you are the most outdoorsy, you've probably all at least been familiar with the process of fishing. If you go fishing, you need basic things. You need a fishing pole, you need the line or the string, and you need a hook, right? Preferably some bait on the hook to attract the fish, but basically a hook. So when you want to catch some fish, you cast your line, right? So you release out the line into the water, and you hope that the hook actually hooks on to a fish. And then once it hooks onto a fish, then you can pull in the fishing line, reel in the fish by getting the string to come towards your fishing pole, right? And that's exactly what's going to happen here. So in metaphase, the centrioles are on either side of the poles. The chromosomes are all lined up in the middle. The spindle fibers are going to be released out of the centrioles. So the centrioles are like the fishing pole. The spindle fibers are like the fishing line. They're going to be released out, and they're going to hook on to each of the sister chromatids on a chromosome. So one centriole is going to hook onto this one on the left. The other centriole is going to hook onto this one on the right. So each one of these chromatids is hooked on to a different centriole because of their spindle fibers. And then here in the next phase, what we call anaphase, both of those centrioles are basically going to pull. And when they both pull at the same time, it's going to break that protein that was holding together the two chromatids. It's going to break that centromere. And by breaking the centromere, you now have two sister chromatids that are completely identical to each other being pulled in opposite directions. This is what we consider anaphase. So in anaphase, the chromosomes that have lined up in the middle are now going to evenly separate so that one identical line moves to the left and one identical line moves to the right. And this will happen 45 more times so that every single segment of the human DNA line gets an identical copy to one side of the cell and an identical copy to another. It's just like a wishbone on Thanksgiving. You're pulling at the same time, those segments will separate out evenly and move to opposite ends of the cell. So this happens in anaphase. So in anaphase, we have an even separation. So identical information is now moving to either side of the cell. And remember, since you've doubled everything initially here, when it splits, everything goes back to their normal numbers. So this is how we're going to actually end up with two cells that are identical to each other and identical to the original cell. So the last basic steps here are pretty much just going to be preparing these two new cells to go off and live their own independent cycle. So the genetic information, now that it's separated out, is super vulnerable. And just like we talked about before, a eukaryotic cell has way too much activity to let genetic information just sit around in the cell free floating. So the last phase of mitosis is going to be something called telophase. And in telophase, a new nuclear envelope, a new nucleus, is going to start to form around both of these clusters of DNA that have started to form. So because anaphase split all your DNA into two even segments, in telophase, a new nucleus is going to form around each one of those. So in telophase, you now have two new nuclei forming around the two clusters of DNA. The very last step of the cell cycle, and this is actually after mitosis, is a process called cytokinesis. So mitosis itself actually has four phases: prophase, metaphase, anaphase, and telophase. PMAT is one way to remember it. Now once you have telophase completed, the cell hasn't actually split evenly. All you've done is separated the nucleus into two new nuclei. The last step is still going to be to take the whole cell and actually split that into two new cells. And that process is called cytokinesis. In animal cells, this process is pretty straightforward because in animal cells there's a membrane, but there's no cell wall, which means the membrane is super flexible. So what basically happens, and I'll refer, since we're in Texas here, we'll think about a lasso and somebody lassoing a calf or something, right? So you shoot a lasso around the calf's neck, and when you pull, the lasso closes. It's the same basic thing here. If you have the cell, you have the nucleus separated on either side, all you're really going to start to do is get these series of fibers around the cell membrane and just start squeezing the fibers. And as they squeeze and squeeze the fibers, it's going to eventually get to the point where they meet in the middle, basically cut the cell into two, and those two new parts will now become your two new functioning cells that each will start their own cell cycle. When you're pinching in on two ends like this, the space that you're developing between two structures, or the partition that develops between two structures, is naturally called cleavage. It actually is called cleavage. So you're developing a cleavage here in the middle of this animal cell that's going to eventually split into two new cells. So I'll let you pause here for the immature laughing, and then whenever you're ready you can come back to play. But that's what cleavage is. It's a partition between two structures. And just like what's happening here, it's going to cleave until it meets in the middle and separates into two new cells. So that's an animal cell that goes through this process because it just kind of pinches both ends. Since the membrane is super flexible and there's no cell wall, you can pretty much just squeeze both ends of the membrane until they meet in the middle. That splits into two new cells. And in this process, of course, you've made sure that all of the organelles have split evenly as well. Mitochondria, chloroplast, endoplasmic reticulums, all of those things have split evenly. So you now have functioning cells. When they split, they can go off and live their own life. Plant cells work a little differently because they have a cell wall. A cell wall, unfortunately for a plant, at least in this case, makes it to where they're not nearly as flexible. It's good in the case that it gives a lot more structure, because a lot of plants don't have a skeletal system, so they need a lot more structural support. But in this process, you can't just kind of pinch both ends of a cell wall, because the cell wall is super rigid. It's not going to bend like that. So instead of trying to pinch both ends and doing this cleaving that you saw happening with the animal cell, everything's going to happen from within. So a bunch of cellulose is going to start to develop, which remember is just the long starch chains that are made when plants have excess sugar and they make this long chain. Plants make cellulose, and that cellulose is what makes up the cell walls. So in this very last part of cytokinesis, cellulose will start to build up in the very middle of the cell after telophase, where the two new nuclei have set up, and the cellulose will start to develop this boundary, a new boundary that will eventually bridge and meet both ends of the original cell wall. That boundary that starts to develop in the middle is something called a cell plate. So in a plant cell, during cytokinesis, a cell plate will develop until it reaches both ends of the original cell wall. That'll become the new boundary line, so that the cells can then split and become two individual plant cells that can each go and perform their function. So one way or another, cytokinesis is the final separation that happens in the cell after mitosis has already occurred. After you've already separated one nucleus into two, which is what mitosis is, cytokinesis then allows for the final separation of the cell. So technically, the very last part of the cell cycle is actually cytokinesis. So mitosis splits the nucleus, cytokinesis makes the two new cells. And now each one of those new cells can go off and function properly. This is what happens in a normal cell. And a normal cell is able to control the rate at which the cycle passes so that it's a consistent amount. Cells are dividing as fast as they're supposed to in order to basically replenish the number of cells that have died off in the process. Basically, it's all about regulation. So in a typical example, a cell cycle and its pace is determined entirely by how often cells tend to break down. So certain types of cells tend to break down within a certain time frame. So the cell cycle has evolved to become about the right pace so that as certain cells die off, you have just as many new cells developing at the same time. So overall, you have a consistent number of cells all doing the same thing. Again, homeostasis is critical here. You always want to maintain the basic same scenario. So you always want about the same number of cells. So if you have a certain number of cells that die off in a time frame, the cell cycle should be just the right rate so that just as many new cells are developing in the process to keep things consistent. There are a lot of regulating factors that go to play here in the cell cycle, all at different checkpoints along the way to make sure that the pace is consistent. And a lot of times for certain cells that have a very long cell cycle, to make sure they don't shoot through G1 phase too quickly, they move off into this other phase within G1 called G0. G0 is sort of like a stagnant phase, basically putting the cell cycle on pause so that it makes sure that it doesn't produce cells faster than they're dying off in their natural environment. So a great example are neurons, your brain cells. Your brain cells are very, very efficient, and they work for a very, very long time. So it's not common that you have brain cells dying out very quickly. And so your cells have evolved, specifically these neurons, these brain cells, so that their cycle is super, super slow. And to do that, they sit in that G0 phase for a long time. Most of the process of the cell cycle, they just shoot out there in the G0 phase, and that way the cell is not going to continue on with the cycle, because the cells have learned exactly how quickly the cell cycle is supposed to happen so that they're able to replenish just enough cells as what are being broken down so that everything stays consistent. Now, once that regulation begins to lose control And once that regulation is lost in a cell cycle, you now have a situation where a cycle can start shooting way too quickly through itself. So it can start going through the phase too quickly. So again, the same scenario, you have a certain number of cells that die off. If the cell cycle has lost its control, has lost its regulation, and now is going through way too quickly, then instead of just producing the exact number of cells you need to keep things consistent, you start producing way more cells than you're supposed to for the number of cells that are dying down. So what you're left with now is a big cluster of cells where you're not supposed to have so many, because the cell cycle has lost its regulatory control. Once a cell has lost its process of regulation, it's going through the cycle too quickly. Maybe cells aren't going to the G0 phase when they're supposed to. They just quickly shoot straight through and continue on with G1 and go through the cycle very fast. You now are making too many cells for any given area. And when you get too many cells in a given area, especially if they're close to the surface, all of those cells have nowhere to go but up, and so you start seeing this bump, right? And that bump, by definition, is what we call a tumor. All right, so a tumor is nothing but an excess production of cells in any given area. And most of the time, tumors are caused by loss of the regulation of this cell cycle. So the cell cycle is going too quickly, producing cells faster than they're dying off, and therefore starting to accumulate in a given area. So a tumor is not always a horrible diagnosis. Most of the time tumors are harmless. Most of the time tumors are not life-threatening. There are situations based on where they are and based on how big they get where they can start causing some pretty big medical problems. But a lot of times, if there's a tumor and a doctor is able to identify it, they'll do one of two things. They'll either go ahead and try to take it out just to avoid any future problems, or they may just say they keep an eye on it and just kind of keep observing it year after year. They keep checking on it and making sure it's not getting too much bigger or causing any kind of a serious problem. But most of the time, tumors are not what we consider life-threatening. And that's because most of the time the tumor stays in one place. So if we consider a tumor to be staying in one place, doctors call this a benign tumor. So if the tumor is benign, that's a good diagnosis because that tumor isn't going anywhere, at least not yet, which means as long as it's under control where it is, everything seems to be fine. No serious problem. The problem is going to come when these cells that have lost their regulatory control, they're going through the cycle too quickly, now no longer stay where they were, but actually start to break off of their own cluster and start moving into your bloodstream. And when they start moving into your bloodstream, they'll pass through and then maybe attach somewhere else, further along in your body. And since they are cells that no longer have any sort of regulation, once they've attached to a given place, they'll start to develop a new tumor in that place. And then again, parts of that will break off and continue to go through the bloodstream and develop a new tumor somewhere else. These tumors that are moving are called malignant tumors. When you're diagnosed with a malignant tumor, you're now diagnosed with a cancer. So cancer, by definition, is a malignant tumor, which is a collection of cells that have lost their regulatory control that are now moving from one cluster to another, from one part of the body to another. And once you have this kind of situation, this is a very serious and very dangerous situation, because now it's not just about one tumor that you try to keep under control. Now it's about new tumors developing all over your body, and each one of those tumors can start to become a very serious problem. And depending on where those tumors start to develop, if they start happening within vital organs and blocking certain pathways, keeping things from happening, you can see how very bad this can get very quickly. So as you can imagine, the majority of the cancer research that's being done out there is all based on this cell cycle and focusing in on what those regulatory factors are, what all the different checkpoints are that happen within a typical cell cycle, figuring out exactly what goes wrong that causes these little changes. And most of the time, these big problems with tumors and malignant tumors all originate from a little tiny mutation. Just one little change in a certain protein that's supposed to keep track of a cell cycle, is supposed to keep it sitting in G0 for a while. Just because of that slight little mutation, now the cycle is losing its G0 property and is going way too quickly. And just that one little mutation could lead to that cell losing its regulation process, turning into a cluster of a tumor, that tumor starting to break off and start to move, and now we can get into a whole different process of cancer and a very serious problem. all because of a little mutation. That's what cancer research is really focused in on, is how to identify these mutations, how to prevent those mutations, or at least lower the chances of those mutations occurring. This is where a lot of our medical research is coming in. These are where a lot of the huge discoveries in cancer research are happening, is understanding exactly what you're looking at here with this cell cycle. Take in all this information. It's very important you have a good understanding of mitosis, because the next unit will focus into the exact opposite process of mitosis known as meiosis, and how it leads to the overall development of future generations through sexual reproduction. I hope you enjoyed the lecture, I hope you enjoyed the unit, and I will see you next time.